10 to 20 times larger than the average virus.
And that represents the amount of life.
Larger than the virus, which is like the biggest part.
No, I think that was one of the, they call it the it's often called chili.
Right. So it's like how much weight on the alcohol
All right, who worked through this before I try to find all the files? Who worked through this? Did anybody work through this? You had a month.
Yes, we have other stuff too.
What? You have other stuff too? I don't believe it.
Yeah, no, I like the kid who can do his homework.
Sorry about that.
I attempted to work through it.
Okay, well maybe we won't go through it because going through everything in detail might be, if nobody has, well, what?
No, I attempted to work through it.
Did you?
Sure, yeah.
I know you did. Okay.
But I mean, I didn't spend as much time on it as I needed to fully understand it.
Well, let's go through some of it, like, conceptually.
Yeah, absolutely.
Everything's online.
Hey guys, this is Roger. Can you hear me?
Hey, Roger.
Roger.
Hey, if you guys don't mind, Cameron had asked that I record this. He's not able to attend unless he showed up there.
No, forget it. He just.
Okay.
I can record if you want, right?
Around.
I have the recording
I know.
set up here, so I can record it.
Okay.
Oh,
So it's okay.
yeah, let's go. Okay, folks. This part started. I have to refresh myself. And I want you guys to work through this with me on the board when we get there. Let's just pull up if the assignment was to do something and I need to work on. Yeah.
I got the assignment here if you want to.
Oh, I have it on my screen.
Oh, you do? Okay.
Yeah. But thank you.
Yeah.
Okay, so the assignment was to, let's just start with the alpha, let's pull up with it, the alpha level. So,
Maybe we should turn the lights off.
I'm going to highlight B and B1. And then there's this region in between. So what is the whole purpose of what's going on here? Like, what is the step before this? What are we doing? Like, what is our hope? What do we eventually want to put in these vectors? Like, eventually, what are we putting in, like, an alpha level vector?
Your gene of interest?
Okay, your gene of interest.
What's
So,
operandi or something?
okay, so maybe, like, you would have this coding vector, but maybe in the place of GFP, you'd want to put your own transcriptional unit, right?
Yeah, so it could be multiple things. Then.
Exactly. Like in here would go a promoter and maybe an RBS and your gene and a terminator. So this whole unit would eventually go into there, if that makes sense.
Yeah.
And then how are you putting these pieces together, which is talked about later in the assignment. But how would you assemble these pieces to go into something like this?
That would be GoldenBraid. Right? You assemble them with the double restriction PLoS.
Okay, so actually, this would be more for putting together like multiple transcriptional units. So how would you put together? So like, let's say in your collection of items, you had a plasmid that carried a promoter that you wanted.
Okay, yeah, you PCR it out.
Okay, you PCR it out, and what would you? How would you? If you're taking a promoter from one plasmid, and then you're taking an RBS gene from another plasmid, and maybe a terminator from another plasmid, how would you get all of these things to assemble together in one pod? Forget this vector, just these pieces. How would you get them to come together? How would you design these primers? What would you do?
Complement sticky ends on pieces.
Yeah, complementary sticky ends. So this is what was talked about at the very end of this assignment, was like, how would you build a transcriptional unit to put into here? And that would be, you would design it so that you would have complementary sticky ends. So you would have, when you amplify up your promoter, you would have your promoter here, but you would have a BSA1 site and a BSA1 site on either end of the promoter. So the BSA1 site would cut this way, right? It would cut inwards, like that, and it would leave a sticky end here and here. So whatever sticky end it left here would have to be complementary to, let's say your RBS gene happened to be one piece, right? Then that would have to be complementary to whatever sticky end was left here after your BSA1 digest. Does that make sense? And it would just go on and on, right? Like this end would be complementary to a terminator here. What did I do? X1 would be complementary to a terminator here. And then this sticky end that is left would be complementary to that vector, right? Like this sticky end that's here is complementary to the sticky end that's left in the vector on the 5' side, and then this sticky end is complementary to the 3' sticky side. Does that sort of kind of make more sense?
RBS is what in this again?
So ribosome binding site for translation. So that was the end challenge was for you to design a transcriptional unit that would actually go into one of these vectors that you designed. And that's something we can still, if someone has examples of that or we want to go through that, we can definitely do that. But is that kind of, does that clear up a little bit?
Yeah, that's good to go through it at the conceptual level like that.
Yeah. So
now that's just using one set of PLoS restriction index. Right?
exactly. So this is all BSA1. So the idea would be you could take your PCR products of all of these, throw them into one pot together with BSA1 and ligase, and it would leave these complementary ends so that all these pieces would align, so that your PR would align with your RBS gene, would align with your TM, which is your terminator, right? And this would all come together to equal one transcriptional unit. So as part of like the project here and the plant bio project is to make, like this is something that we wanted everyone to start thinking about, is what promoters and genes and terminators and all those things do you want to have in our collection? Because that means what we can start doing, and Roger, I'm sure we'll talk about this at some point. I mean, Cameron, they had some ideas about this, is that making like a unifying system where all of our promoters are flanked by the same, like all of our promoters would be flanked by the same complementary ends. And those complement that. So the complementary end on the 3' side of the promoter would align with the complementary end on the 5' side of the gene. And also all of the whole gene collection would have the same complementary end. So like if you could imagine, all of our promoters would be green-blue or something like that. And all of our genes would be blue-red. And all of our terminators would be red purple, and the purple would match up with our transcriptional, or purple would match up with the plasmid. Does that kind of make sense?
Mm-hmm.
So, and by that I just meant this side. So like this end, whatever this end is, that complementary end, would match up with this complementary end. You know, that red one or whatever. Okay, so that's why we really wanted to start thinking like what different little words, or that one of the GoldenBraid 2.0 caused this nice end that I should have done this paper around as well. But it breaks these little components up into like words of a sentence. To think about it in like grammar or language. So these would be like the word, and this makes up your sentence, which is that. And they standardize their words. So we would standardize our words as well, our parts, you know, our words in our sentence, so that we can always piece together like an adjective with a noun and so on. I'm not doing it justice, but we'll pass it around so you can see. So like we'd always look for both motors pieced together with all of our genes, so we can mix and match and do different things and, you know, build our collection, if that makes sense. So like we would build a sentence and put it into a vector.
So they standardize their 4-unit overhangs, right?
Exactly.
And so we should, are the overhangs that they selected? I mean, some of them have start codons in them and things like that.
I believe so. Yes, their first one has.
Like
I
the
believe
one
that
around the
they're.
promoter.
Yeah.
The start in front of the promoter is the start codon, I think.
Or it might be before the gene.
Before the gene, yeah. I guess.
So yeah, so we could even like, and that's something to think about. We could. That's why I said even in this assignment, use their separate regions, like they're in between regions, because they work that out and that makes sense, and we can use that. Why can't we use that as well, right?
Is there a reason to change it, or should we just go with
There's
this?
really not a reason to change it.
Yeah.
Unless we think of something that might be better, but otherwise there's really not a reason.
Right.
And so this is pretty much, we're going to have, that's why let's just focus on the alpha vectors for now. And this is what we'll piece together so that we can make the transcription unit. So in order to put this together and put it into a vector to use in the future, we would have to design, you know, some sort of alpha level vector. And then eventually, if we don't get to omega, that's fine. But that is we would be using these to thread multiple transcriptional units together, or braid multiple units together. So, and Roger, can you hear me okay? Yeah.
Yes, I can. Yep.
Okay. So, let's go through. Okay, so how did we decide, at the very end of the last time we discussed this, how did we decide to make, and I mean, I know I'm posing this to you guys, how did we decide to make the alpha vector? Do you remember? We like all decided on one method, even though there were a couple that were thrown around that were viable. We decided, and all are acceptable, I just thought I'd focus on one of them for the time being.
Right.
Does anyone remember?
Remember we discussed PCR with overhangs as one of the potential methods. I'm not sure if that's what we think.
Yeah, they both involve that actually.
Colony PCR.
Well, that would be to check our flow.
Well, it checks it right. Take your files.
So I think we decided on doing what they called the domestication,
Oh, right.
and they flanked, I think it was, if someone can refresh my memory, I think on the enzyme they used the third type 2S.
Was it BBS one?
I think it was BBS, but I can't remember. BBS1, that works for me. So what they did, so in this case where I said, let's make this GFP alpha vector, you tell me what you would do, actually. What would you do to make this vector? and we can go in small steps.
Okay.
So were we going to use the BBS1 vector for the overall assembly?
We were.
And then we're going to use the other restriction enzymes to cut up the pieces.
Right. So these restriction sites are strictly for the GoldenBraid transcriptional unit assembly, and then they use a third site to actually make this plasmid. So what you would do is take that plasmid that I provided for you, right? And so let's say the GFP gene is here. So this is 5 prime, 3 prime. And how would you get these restriction sites on either side of this gene?
So first you would select sort of an annealing of 28 overlap of the 20 base pairs to overlap.
Okay, so you would have a forward. So where does the forward primer anneal?
So we're going to cut out the GFP and take the backbone. So we start just upstream of the GFP.
Okay, so you could do it that way, where you cut out the GFP. Okay.
Or maybe
No, that's fine. So there are multiple ways you can do this. So this is what I was thinking, and there are many ways to do this. So to make the GFP with these sites on it, you could.
We're trying to cut out. We're saving the backbone. Like you did the other trends.
Yes. So you want it. All you want to do is put. Yes. You want to save the whole plasmid. You just want to put these sites blank, the GFP.
Yeah.
So you are absolutely correct. So go ahead.
So we'll have the whole plasmid with the sticky ends, and with the sticky ends and without the GFP. And it actually linearized at that point, right?
You could do it that way. You can cut it and start with cut vector to amplify off of. Is that what you're suggesting?
Well, just you have the plasmid, but then you have the primers that would anneal and amplify up the backbone.
So exactly. So you can do that. You can do that without cutting the vector.
Without cutting.
Yeah. So you could take, like, you have this entire plasmid, and you can amplify up GFP. So you would have this forward primer here, and this off of this forward primer is a BSA1 site that goes in this direction, a stuffer region, and a BSMB1 site that goes in this direction.
Oh, okay.
Does that make sense?
Yeah.
And
it's
then-
another thing that you know, that's a good way to do it.
Yeah, you can do it this way. And then they put a third site on there. Let's say it would be BS1 that cuts in here, and there's some sort of stuffer, and this will cut and leave an overhang here, okay? So then their reverse primer, which goes in this direction, would have the same thing hanging off of it, except the BSA1 is going in this direction. There's a stuffer region here, and BSMB1 is going in that direction. And then there is the BS1 that cuts this way. So you have primers that anneal to your gene, right? And amplify it, but it'll amplify it with these additions on the end of it. Does that make sense? So that's piece 1 that you get in your PCR reaction.
So you have BSMD1 there, and then what's the other one on the end? After that?
BBS1?
BBS1?
Yeah.
Okay,
So
what's that one?
BBS1 is another type 2 restriction site,
Uh-huh.
so that you can take this PCR product.
Oh, I see, yeah. So then you can cut it again.
Exactly. So then in this case, when you're amplifying the plasmid, so this is piece 1 that you have. GFP with the modifications on the end, right? Does that make sense? Okay. Piece 2 of your reaction is, so this is going on in one tube. You put this plasmid in a tube with these 2 primers, and it's making this PCR product. Right? Okay. Reaction 2 is this template in another tube with a primer that binds, it's going this way, 5 prime to 3 prime, and it's going this way, and it's amplifying your plasmid. And these primers have, what's the only thing that this primer has to have on the end of it? These primers that are amplifying all these different colors. We have another set of primers in a different reaction that are going to be amplifying the backbone. What do these primers have to have on the end, on the extensions, on the end of them?
Step regions for BBS1.
Yeah, that's it. Just the BBS1, BBS1 that cuts in. Right? So
And,
you're,
how do you decide if it's cut in or out? Is there a rule of thumb on that?
well,
You seem to know that very quickly.
so I think of it this way. BBS1, if you have a BBS1 site that cuts out, then, okay, so let's think of it this way. Let's say this is your PC, let's say this is the GFP product, and you have a BBS1 site here, and a BBS1 site there. If your type 2 restriction enzyme sits here and cuts this way, like if you have more here, then it leaves the BBS1 site intact.
Right.
Right? But if you have a restriction site that sits here and cuts in, it gets rid of this and leaves an overhang.
Ah, okay. So cutting in just leaves the overhang.
Exactly.
Cutting out leaves the
It leaves the overhang, but it also leaves the enzyme site.
BBS one's just another restriction.
Yeah.
So
What was that?
BBS one is just another restriction.
Yep, it's just another type 2 restriction site, just like these two are. BSMB1 and BSA1 are as well. But they're reach over enzymes. So the difference is that a non-reach over restriction enzyme sits on the site and cuts in that site.
And the other one just cut that sits somewhere.
Exactly. So a type 2 restriction enzyme sits at a site and cuts elsewhere.
Nina?
Yeah.
Quick question. So when I did mine, I had 2 primers on one overhang and 2 primers on the other overhang. So there was like a BSA and BBS on the one and a BSM and BBS on the other.
Oh, you could do it that way. So
Is there any reason to do it one way or the other? You did 3 and 1, I did 2 and 2, and I'm just wondering if there's any reason I'd choose either of those two.
So you did.
Your overhang had both the BSM and the BSA
Yeah.
site for the insert part,
Yeah.
and then your vector side only had the BBS.
Correct. Well, the primers on the GFP also have BBS.
Yeah, right. So
Okay.
that's why I was saying there's 3 sites on the primer on the GFP side, and then 1 site on the vector side.
Okay, and then how did you set yours up?
I just did the BBS BSA on one side, like on the GFP, and then the BSM and BBS on the other.
Oh yeah, that's totally acceptable.
Is there a reason I would choose the approach you took, 3 and 1, versus the approach I took, which is 2 and 2, or are they essentially equivalent?
I think it's essentially equivalent. Like for me, this is easier to design. Well, I think they're essentially equivalent. So what Roger said that he did, which is also fine, is that he instead of this BSMB1 site here, he had BSA1, BBS1, and BSA1, BBS1. And then he had his BSMB1 here, and the BSMB1 here, so that when BBS1 cuts in, it leaves BSMB1 overhang, and when BBS1 cuts in, it leaves BSA1 overhang, and those anneal that way. Yeah, I mean, actually, this way might be better if these extension regions are getting too long and it's like reducing the efficiency of your PCR. So either way is, does this make sense? Yes, sort of, kind of. Okay. So what he's saying that he did is, this would just leave a BSA1 site on either side of the GFP, and this would leave whatever the overhang is. I'm not sure. but it would leave an overhang when we cut with BBS1. And then these overhangs match up with the vector that just had BBS1, BBS1, and this has BSMB1. And when this cuts, it leaves the complementary BSMB1. It leaves the complementary overhang. So like this gets cut out, this gets cut out, this gets cut out, this gets cut out, and then it's just this, BSA1, BSMB1. So does that?
So in the example where you just had one on the vector,
Yeah.
what's to stop the vector from just closing?
So, excellent question. So when you cut with BBS1 here, the sticky end that leaves here is not going to be complementary to the sticky end that leaves on this side. So it
Should be
shouldn't.
palindromes.
It shouldn't. No, like because BBS1 doesn't cut within the site. It cuts external to the site. So the sequence that it cuts at in the backbone in the separation that you designed on your primer is going to be different than
I see.
what it cuts here. The sequence is different. So like that's something you designed. The overhang it leaves on this end is complementary to this piece, not to it, not back to the vector. So does this kind of clear that up a little bit? So to design the omega vector, it's the same exact thing. There are also other options to this. Neil, I think, brought up.
It wasn't me. It was Patrick and Derek, I think.
Oh, really?
The new the CRISPR thing.
No, I don't know. The what thing?
The CRISPR thing, right?
Oh, the CRISPR thing. No,
Oh.
I don't know about what's the CRISPR option.
That's not.
That's not for
this.
this?
Is for you can make
To make a genetic mutation?
you can basically if you synthesize a small RNA,
Mm-hmm.
it'll direct double strand break. You can then use the new molecular combination to actually then
Yes. The new George Church wave of genetic
Yes.
engineering. Okay. So, but didn't you bring up like another way to make this plasmid? I thought you may have. Maybe I'm misremembering. So like another way to make this that would take longer, but it's also a viable option, is you can take your template and it's okay if I erase something, I'll erase this. But I like this method that they use in the paper, this domestication method. But the other option is to, if you have this is your plasmid, and your GSP is here, and you wanted to insert, something in this region, you would have, I'll draw the other screen. You would have a primer that binds, here, and it just has the long extension on it with all three sites that you want, or whatever. And you amplify up the entire vector all the way around. So you have entire plasmid plus these three sites. And then you can take this.
So that would be linear then?
Yeah, you can either take this and transform it into cells that will ligate it and make the double strands. That's one option. Or you can amplify this. You can have the other primer that's going in this direction. So you make a double strand of this and ligate it and transform. So you'll have new plasmid plus region inserted here. If that makes sense.
So if you just had one primer there, and you had those three sets of things at the end, so it'll create a circularized plasmid with the insert?
Yeah, if you had ligate, so put in the cells that can ligate.
I see.
Yeah. So you can.
Nina, does that do a blunt end ligation?
Yeah.
Okay, so then you just want two of the sites in it then, not all three, right?
Oh yeah, I'm sorry. That is correct. Thank you. So you would just have the two sites. Okay, so I would say the more efficient thing to do is have your two sites on a primer that is the forward and or whatever you want, and the other primer is reverse, and you make a double strand. And then you can take this and you ligate it, it's one-ended, and transform it. And then you would have to sequence this region to make sure that this was incorporated. Then you can use that for the template to do again on this one with another set of primers.
That's the domestication approach?
This is not. This is the domestication approach.
Oh, right.
This is just the old-fashioned way. The other option is to put the third site here, BBS1, and also the third site here on this primer. Does this make sense, sort of? When you get this double-stranded piece, you put it in a one-problem BBS1 and ligase, and it cuts and ligates it, and that's more efficient than a blunt end. But then that would still involve you make it, you transform it after you ligase it, you mini prep it from cells, you sequence through this region to make sure your sites were incorporated properly, you pick the clone that it was incorporated properly in, and you use that for the template to engineer the same site on this side. So that is more steps than that, right? So this would be the preferred way. But this is totally viable and will work if for some reason you're doing this and it fails and you want to try another option or whatever. Both of these will work well, but this is probably going to be the more efficient of the two. So that's what's making the alpha vector. So the only thing that you would have to do is make sure that when you're designing the primers to amplify everything, that the restriction sites are going in the appropriate direction.
How difficult is it going to be for us to engineer out existing sites? I think we talked about that at the beginning of the class.
Yeah, so if this plasmid has a BBS1 site in the backbone or BSA1 site or something like that, yeah. So what we would do is, that is another step of amplification and changing the plasmid. So if this is a BSA1 site here, we would have a primer that's complementary, like in this direction, and in this direction, and it would change the site here. It would change like one of the nucleotides. And we would amplify up the whole thing. So it would anneal, but it would change, does that make sense? It would anneal, but it would be off by one nucleotide. Mismatched. And so you want to make sure you change this to, you want to make sure that you change this site to something that keeps the amino acid sequence.
So just more on a practical lab environment kind of perspective. We would have to do PCR for each site we want to remove, and then we have to transform cells, grow up new plasmids. So we have to do that for each and every site, right?
Exactly, yeah.
Okay. Might be easier to just order new plasmids.
Well, yes. It might be easier to acquire plasmids from someone that has already mutated these sites or find plasmids that do not have these sites, or the other option is to pick our enzyme choices so that we minimize the amount of times we would have to change the existing vector.
So use different textures.
Exactly.
That's some of them are sort of expensive though.
Yeah, they are.
But you only have to do that once. Once you've got your vector without the site, then it's good for it.
Forever.
Forever, yeah.
Yeah, that is true. So an extra week or two of hard work
In the beginning.
at the beginning.
Pays off, yeah.
Exactly. So some of the vectors, we might be getting some plasmids, I think, from this group that designed GoldenBraid. So hopefully their vectors, knock on something, I'll knock on something too. Their vectors should already be engineered to not have these sites in the background.
That would be handy.
Which is awesome.
Yeah. It's so wrong.
So then all that you guys would have to focus on, if you're using the plasmid for plant bio or for something else, all you would have to focus on are the parts that you want to put in STEM. Like designing those parts that fit in the vectors that they give us. And then that's also something that's like we're hoping to decide upon is like employing and Roger can speak about that. Roger's going after this, I think, right? Designing like complementary ends and enzymes to pick and stuff like that, right, Roger?
Right. Yep.
Yeah. So once that gets all sorted out, and I'm sure if you guys have any suggestions, right, on like how to make complementary ends or what ends to pick, I think we might just stick with what the GoldenBraid is already done, and then we don't have to think too hard, and that has already been worked out.
If it's a working system,
Yeah.
I mean, might as well just let it out.
Yeah.
Do they have bacterial vectors or most of the stuff plant vectors?
They don't. They just have plant vectors. So this is an opportunity for us to engineer bacterial ones, and I don't think they're interested in doing that so much.
I think that would be a good project for us.
Yeah. And then we can try to make all the parts like work for both vectors, right? Like work for all sets of vectors. So if we have insect backbone, I don't know, or subflora bacteria, and also for plants, then we can make sure all parts fit all those backbones.
Mm-hmm. Are there bacterial backbones that you suggest we do?
I mean, I think the standard, like the cup, is pretty good, and I don't know. I think that's crazy. A lot of the backbones that we have in the lab are like weird combinations of.
The PBAD one is
Yeah, no, PBAD's good.
PBAD, okay.
Yeah, that's good. I think what we have is really good, so we can just engineer hopefully what we have for the E. coli system.
Okay.
So it would literally be exactly what we just did, putting these sites in the pico vector, and then making sure that we mutated these sites out of the pico vector. That's a project. I mean, we can get started on this like immediately. I think we have a clear idea of how to make these vectors and what we have to do.
We want to replace GFP. We don't want to leave the GFP in there, right?
No, I think we should leave the GFP in, because then if we're putting a transcriptional unit in,
so we'll have a marker.
Yeah, exactly. Like all of our colonies that have our transcriptional unit should be white, and then all those that still have our background would be green. And I think it would be nice to keep those. So I think we should literally take pico and put these on either side of it, and then what the other plasmid is lacking. Is that why I chose that one? What else we have? The cup 18 is lacking.
Yeah.
So then maybe we should just engineer that to be the omega, and then put these sites on either side. So what happens here is that let's say you engineered a transcriptional unit, and your transcriptional unit now is flanked by complementary ends that have been cut with BSA1 sites. Well, now this vector codes BSA1 sites too, and match up with these complementary ends in the red boxes, so that your transcriptional unit fits in there. And then so you have multiple plasmids with transcriptional units in, right? So then you can cut with BSMB1 and cut out all those transcriptional units and put them together in the omega petri ball.
Keep going back.
They keep going back and forth.
I have a question for everyone.
Yeah.
Did anyone actually go out and design primers? Like, did anyone go that far?
I bet, Roger, did you do that?
Yes, I did.
Are they on something that I could share with the room?
They're in a Snap Gene document.
Well, that's cool. You just email it to her.
Yeah.
Okay.
Sure.
Okay. Also, Snap Gene has kind of a cool thing where you can actually run the PCR in it, which is nice.
Cool.
Do you have the viewer, Roger, or do you have the app? Yeah.
I got the app for 30 days, and then I don't know what I'm going to do.
Oh, okay.
Because I'm not paying $500.
Yeah. But does this kind of clear up like what has how to design these and what has to be done. And then if you guys actually sit down and make primers, and I think Roger has, obviously he has, we can go through them and check, and we can just start ordering them and playing around.
So, did you order any from IDT, Roger?
Oh, no, I didn't actually make physical ones. I just designed them.
Oh, okay.
Was that what the question was? Did I actually order them?
Yeah, we wondered if you'd ordered it, or I wondered if you ordered it.
Oh, no.
Yeah, okay.
What haven't you done the reaction yet?
It needs to be an Android version of Citation.
What was that?
An Android version.
This is like.
We just won a free tablet,
Oh yeah, I heard about that. That's awesome.
so we could kind of buy one to dedicate all pitching for one copy of Citation. But we don't have a computer to put it on the size of contest. But a tablet.
I wonder if we approached, it sounded like they suggested for labs and, students working in labs that the lab buy one and the students just use it. I wonder if they'd extend kind of the same philosophy to BioCurious.
Oh, I'm sure they would.
I think they would, yeah. Maybe I'll call them and ask them.
Yeah.
I'll do that. That would be useful, I think. Do you find the app pretty useful, Roger?
Yeah, it had a few things like being able to, you could run primers on it, run a PCR. I think that wasn't innate, if I remember, or Nina, maybe you can correct me on that. They also had more up-to-date enzymes and everything more complete, it seemed like.
You guys use that in your lab, Nina?
We use 8.
Or use 8, okay.
But I do like, I don't know, Snapchat is really aesthetically pleasing. There's something about it that. 8 is pretty minimalist.
Yeah.
Yeah.
Just spell
8? Yeah, 88, yeah. It's capital A, lowercase. It's A plasmid editor. That's what it is.
How much does it cost?
8?
8 is
It's
free.
free.
But Snapchat, how much is that?
Yours is free.
Yeah.
The viewer is free, and there's a trial for the full version.
That's what, $500 a month?
It's like $395.
Roger, are you going to put it in the Dropbox or send it in the email?
I'll send it through the email first here since that's the direction I was going, but I can drop it in the Dropbox if you have that also.
Oh, no, the email's fine.
I actually want to try that. I mean, I'm kind of halfway through, but I wanted to make my own primers and see if they work. Would anyone be willing to read my?
Yeah.
Alright.
What's more practice?
Huh?
What's this sector?
Designing
idea.
primers
Came
for
with
this
that
whole GoldenBraid.
Oh, okay.
I'd be interested, so I need help with plan. I need to
I
practice
don't know.
primer
It looks
design.
nice, though.
Yeah.
I mean,
EGFD.
like someone
Yeah.
who knows how to. Like, we can work on it together.
Yeah.
I was thinking someone who actually knows what we're doing. Like me.
Do I know what we're doing?
Yes, we all know what we're
The
doing.
blind, doing the blind. Except for Roger.
No way. Hey, don't you think you guys are much better equipped now to design primers?
Oh, yeah.
There was a cool
Art and
exercise
design.
that I.
Yeah. I'll put some of the fun out of that for the primer pen for bidding.
Oh, that sounds good. I like that now we're just giving out
You're
homework.
going to be dragged into this.
Yeah, this
Primer.
is
You guys didn't know you'd be signing up for our classroom homework.
Oh, it's good.
Yeah, that works. New things like this is primary things fly out the base
Hey, did you guys get little Illumina USB drives?
We
You got one?
We actually did get a IC.
We did. Did you get one?
Well, we got 2 sequences. I went over and picked them up.
Oh, those old sequencers?
Those old ones, yeah.
Yeah.
They were giant.
You
They weigh
guys gonna
about
take
20.
them apart?
Yeah.
Yeah. Pretty cool.
I think everyone just admitted some of that.
Yeah.
Hey, Nina, those have been in the mail for a bit, so if you want to check, hopefully they're there.
Oh, yep. Okay, cool. Oh, good. Did you attach that paper?
Yeah, I think I did. I threw in the GB2.
I'll show that really quickly, actually, because I didn't do a good job explaining it, and I'll send it to you guys. But Roger found this paper. So GoldenBraid 2.0 is like their next generation of GoldenBraid, and it's practically the same thing. But they just break things down to you in a very illustrative way, and they just, talk about, instead, they broke it down into more components, not just the promoter and the RBS and the G and the terminator, but they break it down to other regions, and it's kind of nice that they break the sentence down into this part. So they're trying to relate to the transcriptional unit into grammar, and I think that helps, right? This is the whole terminator region, and this is the transcribed G region, and this is the terminator. I don't know what I just said, but this is the whole transcriptional unit. And they go through and describe these components and parts before getting into the nitty-gritty of the science of it. So I'll pass this paper around so that you guys
that'll be
can
great.
read it. Yeah.
Great.
You'll send us all email?
Yeah.
I like the analogy that they made with the grammar, breaking it down into a sentence is noun, verb, noun type of thing. And they did their grammar, their overhangs are kind of equivalent in a transcription unit way.
Yeah. They go through, they really show exactly how the enzymes are cutting, and what direction, and they show you their stepper region, so you can see that the BSMB1 site is cutting within the nucleotide and what overhang that means. And their PDGB is their name from GoldenBraid Omega. So this is how the omega works, and this is spectinomycin resistance on their backbone. And so it's kind of it's a really nice descriptive way of going through everything, I think. I don't know, it helps. It tells you, so it's giving you there are two options for creating, putting their words together into one transcription unit. So this is the option A is what was asked in the assignment, the last thing in the assignment was putting BSA1 sites on either end of your word or piece and having them come together to form one transcriptional unit. And so it is nice. They go through and show that, and then they actually have another option with different enzymes and, I don't know. It's just a nice descriptive how BSA1 site is cutting and all these.
This is for the reason paper? Or?
This is, I guess it's a manual. I don't know.
Oh, this is a user manual. Wow.
I think this is unpublished right now, isn't it?
Published,
really?
It was published to me.
Does it?
Wait, did they send this to Cameron?
Is that the URL for it right there?
I guess so.
It says user's manual up top.
So I think it's worth going through that. And,
Oh, yeah.
I mean, I can send it around. This wasn't something that they just sent to Cameron, right? Or is it? They don't. Do they care? You think if we just get sent around?
I don't think so. They did send it to Cameron, but I don't think he had any don't forward
Yeah, there.
instructions.
They showed the part.
Yeah. Okay. Awesome.
If you could send it, that would be great.
Yeah, I will send it to all of you.
Are there any labs that you know of that are using GoldenBraid?
I don't know anyone using GoldenBraid.
Okay. But just because it's fairly new, or
I think
are there other things that are better? Or?
I don't know. What are the plant? I think Kyle was saying that in the plant groups mostly they just find ways to you do enzyme digest and ligate pieces together without this type of one pod, this type 2 restriction enzyme scenario.
Yeah.
So
you go gateway is another way that it's done.
yeah.
Most people were off the top of the column back then.
Mm-hmm.
Nuclease systems. It's just kind of come out of the plant bio world. Not really new. Kind of a way of try to get the front document as well and think more broadly in terms of psychology.
It seems also to be a great way to standardize stuff and share, parts and things.
Yeah, absolutely. Okay, what did I do? What did I do with Roger's stuff? Okay, ego primers. So, do you want to go through what you did a little bit, or would you like me to?
Let me pull it back up. I mean, you've already described what we did. It's just, I did the same approach that you just described, except I only used 2 restriction sites on both.
Okay, so, well, I guess I have a question. Does Snapseed, if you give it, so you can add features to it, right, to highlight? Okay.
Yeah.
So that might be beneficial too for you guys to add these sites as features into your DNA libraries on whether you use A or Snapseed, because then it'll highlight where the restriction site is.
The version I have automatically detects using websites.
Oh, does it? Okay. That's good. And then you can also set it. You can set it so, if your restriction insights, if your site's going 5' to 3', it'll be one color, and if it's the reverse complement of that, it'll be another color. So you know what orientation your site is in. But yeah, it looks like Roger has something that completely anneals to this portion is completely I don't know. And this is an extension. I don't know if I'm reading this right.
Which document are you on?
I'm on pigoprimers.dna. Yeah.
So it looks like that one right there. It looks like it anneals to the top strand.
Yeah, so there's actually, I think, 4 pairs of primers here. So the notation, the first one is it's P-glow, and then it's the A12C part.
Okay.
And then the C is for the cassette forward primer. Does that make sense? Can you see where I'm reading that?
Yeah, so this is...
The very top primer is pglow.a12c.c.f.
Okay.
So that's the primer to get the A12C part.
Okay.
It's the cassette primer, and it's the forward. So it's the part that's going to pull pglow out.
So this part should be what Roger's saying is that this is GFP. Then this primer is annealing to here and going in this direction, and it has his BBS1, BSA1 there. So that's the forward.
Right. And then the other one on the top strand is the same, except it's the C12B primer.
So there were 2 types of. So if you guys weren't in the annotation of the paper, there were 2 different types of alpha level vectors, and so those are forwards for each alpha vector that they made. So Roger designed primers for both of those alpha level vectors. Okay.
And then the lower strand, the notation there, again, it's the pglow plasmid A12 part. This is the vector primer, and it's the reverse one.
Okay.
So I call it reverse because it's on the bottom strand.
And
See how in the portion of the primer that anneals to the plasmid, Roger, how many bases did you use for that?
So one of the things that was kind of nice about SnapGene is it would give you the melting temperature as you added primers or added bases. And so I designed them all for 60C.
16C?
60C.
60, okay. So you added, so the annealing overhang is based on getting the right melting temperature.
Right.
Right, I got you.
So the only part that anneals has to have the right bonding temperature. Yeah. Is that what you said?
Yeah, I was just curious.
Yeah.
Yeah.
So this, I'm sorry, I missed it. So this reverse primer is for the vector. That's what the V stands for.
Correct.
So I'm sorry, I drew this wrong. That's not what this primer is. The other reverse primer, the other primer is for the vector. And that is also BBS1 and BSMB. So like that would cut this way and this would cut this way. Yeah. Okay, cool.
And so just make sure it's clear. So there's 4 sets of primers, 2 for the P-Glow that'll fit into an A12C part, and another P-Glow that'll fit in a C12B part. And then there's 2 more sets of primers that'll generate an A12C vector and also a C12B vector.
Okay.
And I don't know, Ian, if you know a way to do this, but one of the things that SnapGene didn't seem to do is it won't show me the recognition sites in the primers. It'll only show it in the main DNA strand.
It won't show you the the part that anneals?
Well, I guess the anneal part you would see because it matches the main DNA, but the part that's the tail that's hanging out there,
Yeah.
it won't show me that there's a BSM or a BSA site out there, or BBSM.
That's what I was. So yeah, that's what I was wondering if you could do that. Because like in 8, if you put this, so this is an example. Let me copy this and put it into 8.
Yeah, actually, able to read the DNA file.
And then it'll search this for features, and it should highlight. Like it's not going to highlight everything because they don't have. Well, there's an X bar right there. Okay. I don't have. So it's not picking up a BSA1 thing. I don't know why. And I don't happen to have BSMP1. I don't have those restriction sites in my feature library, but you can add them to this, and then it should highlight those restriction sites within the primer. Oh, maybe because these are ends here. Is there a reason why these are ends?
Oh, I had a couple N's in there. Just those don't matter. Those are outside of the recognition site.
Okay. So yeah, I don't know why.
But you can, if you open up the PGLOW, like PCR, PGLOW A12C.C.DNA, I think I included a couple of those that were PCR products.
I see a GGAT in the purple text here, but I didn't see it in the 8 basic section. I don't know why. Maybe I just missed it.
You can add all those. So this would be PGLOW, PCR of PGLOW A12C DNA. So this primer is the forward of the insert there. Okay, I don't know how this is set up. Okay, so this is the PCR, and then what do I have to do from here?
Well, now you should be able to see the BBS and the BSA sites up at the top there. See how they're highlighted?
Yeah.
If you hover on those now, it'll, because those are in the main DNA, it'll identify them.
I see. Oh, I see. So if you're adding it, it's not. So I wonder if
So it lets you have PCR primers in the file.
Yeah.
Somehow it'll display them at the same time, but it doesn't run the same recognition site algorithm.
I wonder why that is.
If you try to label them manually by hitting Control T, does that work?
I don't know. Oops. So Control D.
Yeah, just select the section of DNA you want to label. So you can call it G8 sequence.
Yeah, I tried that. It shows it up as a feature, but it doesn't hover above like the other restriction enzyme sites.
Oh, okay.
Anyhow,
I don't know.
that was just a little bit of a nuisance, but it was not. It was nice to be able to run the PCR and see that the products work the way I expected.
And it's nice because this tells you, like when I like this a lot, because when you hover, and 8 doesn't do this, when you hover over this double strand, it shows you that BSA1 is sitting here and cutting this way.
Right.
Like the way the site is oriented, it's sitting here and it's cutting one away here, and it's going to leave this overhang, where BDS1 is sitting here and cutting in, which you want. So that is really nice with matching. So, but you have to, as Roger saying, you have to design your primers and run the PCR and get the product in order to see to make sure that your enzyme sites are in the right direction. And cutting them right.
And Nina, I don't know if you've looked at the actions. I haven't tried all these, but
I haven't either.
it has some pretty cool. It seems like pretty cool things that you can do automatically. So it's like it's got the mutagenesis in there. It's got destroying restriction sites.
So Roger, do you think you could send that file out to the group with your primers on it?
Oh yeah, I'd be happy to.
That would be great. Yeah.
I'll send those and the ones with the PCR results too.
Oh yeah, that'd be cool.
So, what do we think? We want to all go home and like design them with the actual nucleotide sequence and all come to a consensus and order primers for here, and then Roger can have his primers in Pennsylvania.
So
We can
this
have a
one still
primer
has a
off.
few ends in it here. We just fill those in with whatever we want. Is there any rule of thumb on what you want to replace those with, like C's and D's? Or?
So why? Was there a reason why you had ends in these locations?
Only that when I was typing in what the primers were, it didn't matter what was at those locations because they weren't recognition sites.
So the only thing I would say is when you guys are designing the primers, you just obviously want to make sure that you're not like creating an unnecessary restriction site if you don't want it there, right? So like, fill it in, and then also this region doesn't matter so much with regards to melting temperature because it's not annealing to the template. So I would say if you could fill, if that region was ended with annealing to the template, I'd want to make sure that I was replacing it. I was balancing out the ends. Like if I had 3 spots or 4 spots, I'd want to evenly distribute these and G's. Unless I needed to increase the melting temperature, I would add G's or C's. If I needed to decrease the melting temperature, I'd add A's or T's.
Do we want a smaller annealing temperature for the overhangs?
You don't need to worry about the annealing temperature of the overhangs at all. But what you do need to worry about also is take your overhang, just the overhang, and make sure it's not complementary to anything else in your plasmid. Because if it is, then you're going to get like mispriming. Like your overhang will anneal to your plasmid and try to amplify some other region. So it's also important when you're adding these long extensions on the end of your primers, and it shouldn't, but if your backbone happens to have BSA1 sites in it and it's flanked by something that's similar to what you have in your extension, it's possible that you could have some
We want to get rid of those anyway. So.
You do, that is true, yeah. So, that's always just a good rule of thumb. If you're putting a long extension or any sort of extension on the end of a primer, you just want to make sure that it won't yield to any other region of your vector. So that might help you decide what you want to put in those end locations as well. So I think we could probably, we could divide up paths too. I think it's a good exercise for everyone to go through designing the primers and cross-checking with each other, probably, like checking each other's designs and learning from them. And like we can just have a group, I guess, and send out each other's designs and take a look at them and compare yours to whatever, see. And then also, if someone wants to take on looking up, checking the backbones and seeing how many sites we have to change and how the best way to go about doing that, maybe. And so we could have like two. So all the weight doesn't fall on one person. It would be nice to have a few people maybe focusing on like modifying the backbone, like ordering the primers for that and designing the primers for that, and then. I don't know. That's totally your call. How do you want to divide up the work? What do you think? I don't know. Or maybe not. Does everyone want to try to design everything? I don't know what you want to know.
I agree. I was actually going to suggest that, but then when Roger said he finished everything, I felt like, oh crap, I got to learn how to do it all on my own too.
Everyone's competing against Roger.
Here's an idea. Why don't we prioritize different pieces for each subgroup or each individual, and that way we'll have everything done as a group, but we can keep going, on the back burner on our own to figure out how we want to design practice for everything else.
Yeah. So the only thing is that when it comes time to actually starting the PCRs, you obviously want to modify the backbone that gets passed on to the people that are going to be putting the enzyme sites in, right? Or you want to put the people putting the enzyme sites in or passing it on to others that are trying to mutate the site out of the backbone, one or the other. So those, when it comes to experimental, that shouldn't be two independent things. That should be like coordinated step one, phase one, phase two, or something like that. But I do think that there are three independent things that could happen. There could be like the alpha design group, the omega design group, and the design group for mutating the sites out of the backbone.
Right.
Or out of the backbone.
We have to mutate the sites out before we.
You could do it either way. You could put these sites in and then mutate the other sites out,
okay.
Or vice versa.
Of course, we'd have to worry about annealing it.
Yeah, but hopefully, the context of the restriction site in the backbone would be different enough than the restriction site flanking your GSP gene or last gene, that it wouldn't matter. But you are right. That is something we have to take into account. We can decide what is the best way to go about that based on the time of life. I don't know. How do you guys feel about that? Dividing and conquering? You go, yes.
Yeah, I think that's a good way to do it.
Now, does everyone want to participate in this? Or do you want to, you're not up to speed enough? Okay, that's totally, would you like to get up to speed? Or do you not have, you have enough on your plate already at the
I've got a
moment?
lot on my plate, so.
Totally acceptable. Nobody is judging you, don't worry. No, it's totally fine. I completely understand. So how about this? If you're interested in being part of the design process, when we're done or something, do you want to I'll write the 3 categories on the board maybe, or do you want to, and you just write your name under it, or do you want to box? I don't know. I don't want to, if you guys want to think about it and see how, figure out how much you have going on.
We should decide the way I think.
Unless you're ready to make a decision at this very moment.
I'm ready.
You're ready? Okay. Well then, I'll write down who's ready, and those that aren't ready, you can think about it and then decide. But you don't have a lot of time. What about all categories? Yeah.
One suggestion: if you I think it'd be useful to define exactly where you want the inserts to go. So, like for instance, I cut right in front of the ARAC, and I forget where on the end. But if we want to compare our primers to see if we got the same answer, we're going to want to know exactly where we planned on inserting.
Yeah. Yeah, that makes complete sense. So that's something we have to think about actually as an overall design because, and thank you for bringing that up because I wasn't even thinking about that. Like the PLoS vector has a promoter and then the GFP. Now, Roger designed the primers so that the promoter would also be cut out. I mean, the enzyme sites would be flanking the promoter and the end of the gene. Is that right, Roger?
I inserted the two recognition sites between GFP and the MCS on one end, and then on the other end, just in front of the regulatory, is what I assume to be regulatory, ARAC.
Yeah, but like
In front of that or behind it?
I put it right in front of it.
So you took it out basically.
I assumed it wanted to be, part of the cassette, right? So you're going to insert your own regulatory whatever.
Correct.
So we want to take out the operator and the promoter.
Yeah. I can't find my PLoS vector actually. But, so, you want to take out exactly. You want to take out the because we're going to be putting in our own regulatory elements. Right? So you don't want to put the sites here because you're probably going to be putting in a promoter.
Okay.
Right? So you don't need promoter. So the best place to put the sites would be, and also on like backing that up even more on like a more fundamental level. If anytime you put junk in between a promoter and a gene, it totally changes gene expression. So like this is already designed to be promoter and RBS that drive for GFP. So like if you're putting a chunk of nucleotides in here, like that could be disrupting the RBS, it could be disrupting the promoter. So it's usually never, we'll just leave it at that. So like it's not best practice necessarily, unless you have to. So sometimes like you'll have a restriction site in there and it's fine, you tested it and it's okay. And some restriction sites actually like NEG1 codes for a start site in ETG. So like you choose restriction sites wisely like that. But in this scenario, it doesn't make sense to do that, right? So like you're going to want to put your site here so that when you come in with your transcriptional unit is going to be PR, coding region, set, and then terminator. So that's going to go in here. So think about that. So like if you're working on a plasmid that has a terminator already, then you want to put the restriction site on the outside of that terminator. So when you like think about it and get together, if you're going to be working with another person on developing the primers, like think about all of that.
Well, shouldn't we make a decision here though?
Like exactly what nucleotide to start at?
Well, not maybe at the but so basically we decided to cut out the promoter that's in the PDAD plasma
Right.
backbone. Okay.
So would it because like you're just engineering the sites in here? So like when you put this expression vector into cells, it's still going to express pure GFP just fine. But now it becomes a cloning vector where you can clone out that entire PR GFP set and put in your own from different promoter. Like maybe this is PDAD and you want to have CMV, or you want to have the G7 promoter, or you want to have like the PCAT promoter or something, right? So now you won't have PBAD, then PTED, and then your G7.
Right.
So Nina, just point of clarification, you keep using the term promoter, but I actually went further upstream than the promoter up to the what is it, L-arabinose regulatory protein?
Want me to send you the PLoS reference?
So, yeah, that's fine too, but that I'm like a little conflicted on because if we use the PBAD probe, then we're just going to have to clone the regulatory element back in. And that's fine too. but I don't know. Like, I don't know that we need to get rid of RSC. Do we? What do you? I don't.
Pretty big.
I really don't. Is it big?
Pretty big, yeah. You should have it.
If you want, maybe more, maybe you want maybe two different.
Like RSC for all the
right?
PLoS
You
ones?
have, like, maybe you have one version where it's not exactly like the GoldenBraid, but it's like you use the system to cut out GFP and put whatever you in that's been regulated by arachidonics, right? And you have another set that is independently arachidonics. Right, because I can see that arachidonics RSC being another TU, right? In long term, right, you would have RSC, all of that regulatory stuff as one TU. And then whatever you make here using the arachidonics as another TU, you can go with extension.
Like, I don't know what's going to be easier. Like, we leave ours, like, or maybe we just have two backbones, one that doesn't have RSC and then one that does.
Right.
And then we can clone units that aren't PBAD into the one without RSC and the ones that are, because not all the promoters we're working with are going to be regulatory. Like, just TET maybe. Oh, LAC, if we do lactate, like a LAC induction, that's going to be regulatory. And,
Might be handy to have one with PBAT promoter in it. That way you can just throw a gene in and you want to express and use that promoter.
That's what you're probably going to do for Lux, right?
That's what we were planning to do.
Might be
Lux.
separate.
Use that. So Lux uses this quorum sensing promoter mechanism. It's very sort of hard to deal with in the lab. I mean,
so you would want to keep the regulatory elements. Like maybe for your, it's like, I don't know, Roger. I think maybe we should design two.
I actually wasn't sure which was right. I was just saying for this exercise it'd be good for us to pick it so we all end up with the same primers.
if you look at the map view here, you can see the
Yeah, so this is the regular, so Roger designed so this site to be in here. I'm sorry, to be here.
right up there. Yeah.
Up here. Here and here. So the option is to, so the RC actually, if you got, the RC inhibits, blocks this promoter. And when you add arabinose to the media, it binds up RC. Is that right? I forget the whole mechanism. It binds to RC and then releases repression on the promoter.
Yeah.
And so the more arabinose you add, the more expression you get off of the PDAC.
Right.
So you'd always want to have this regulatory element in when you're using the PBAD promoter.
Right.
And if we think that we're going to be using the PBAD promoter a lot, like the benefit of having PBAD is you can do various levels of arabinose, and that means you can get varying degrees of expression off of that promoter. So you could have like low expression in some situations and then high in others, depending on what you're studying. Mm-hmm. So part of me thinks that maybe we should, the alpha people should make a variety with RSC and a variety without. I don't know. What do you think? What's the consensus on that?
I think that's good.
Is that
It's
okay?
not that much more work, right?
Or no?
You do them
Yeah, I think that
simultaneously.
seems good. The other one would be just the one with RC designed to just incorporate that promoter, right? I mean, just
So,
cut out GFP.
yeah.
And you're only worried about your GFP.
So what Bio's suggesting is you would make like, the other option is you would make the P, like if this was PBAD, this PBAD and RFC regulatory element would be flanked by like BSA1 site, would be flanked by the site, and this would be treated as one word of your transcription unit. So when this,
That makes sense.
it would be like regulatory element promoter,
Right.
which is also completely acceptable.
It gives you no flexibility.
And it is 879 base pairs just the RSC, and the p-value is smaller than that, it would be like 1 kb probably-ish, which is totally doable. So what do you guys like more? You like So then let's think bigger picture, like what's going to be easier. So that means that in our parts repository that we make, we're going to have to probably, we want to have a p-value element that's standalone maybe, and a p-value element that's, like a p-value element that's flying by the site, and maybe also RSC p-value that's flying by the site. Just in case.
Well, what's the matter? I mean, hopefully that would be nice, but
Yeah.
we're not talking about it, right? I mean, we can talk about, yeah, we'll make this fun.
Yeah.
And that's good to do at this point. But when we all break to do our, the actual work behind it, we're going to have a lot of other stuff on our as well. I'm thinking we can pick one as the primary.
I think I'd like to design one that does have the promoter, the ARC promoter.
This regulatory element?
Yeah, and then I could
Because you're
pick a
going
Lux.
to use it.
I was going to use it for the Lux Opera.
So the next question is, do you want it so that your plasmid has this in the backbone? Is that easier for you? Or do you want this as one unit? And your plasmid
It would be
is
easier just to have it in plasma, but maybe that's not convenient for other people. I mean, it's not really that much different. The only thing you have to
not
do,
different.
everything's the same except for the annealing part, right? The primer. It's just a matter of where you place the annealing.
I mean, maybe it would be nice to, get rid of the RC, and then we could always have like the 5PTG inducible promoter, and it's like operator regulatory element in front of it as one unit, and then PFET and its regulatory element.
Hey, Nina?
Yeah.
Can you bring up that GoldenBraid 2.0? Because it does have, it talks about this a little bit, or it at least has some graphics to support it.
From a time point.
From a time point, I mean, if we could, I'm just thinking about doing the reaction, right? So if we ordered sets of primers to do all three options, we would just run them in the same, run them in separate tubes, but we could just run the PCR simultaneously, right?
Then we have to do the transformation.
Nina, 2.1 to start.
Oh, 2 point.
We just run everything in parallel. So we run all sets of the experiments
Figure 2.1, sorry.
in parallel, right?
Yeah, you can. It's just more work. I mean, it's just more work, right? It's like more chance to mess stuff up. It's more primers you're handling, more transformations, more,
Yeah.
Which is, but you're right. It can all be done in parallel.
We would just have three situations, right?
Yeah.
Before, after, and flanking.
Yeah. The only thing that would change is you would have a set of primers for this, in front of the promoter, and a set of primers for the RC. Right. It would be another condition.
Yeah.
I have a philosophical question.
Yeah.
Is the whole idea that we can move these parts around? Is that why we are doing GoldenBraid?
Yeah. So like, it's not necessarily move them around in the sense that they're interchangeable.
Yeah.
They're interchangeable. So like the promoter and its regulatory elements are always going to go in the same position, and the coding sequence is always in the same position, and the terminator is always in the same position. It's just a matter of being able to swap them out, like change the gene, change the promoter easily.
So
figure,
I'm sorry, Roger.
I was just going to say figure 2.1 that you have up there now kind of shows the grammatic analogy that they use between sentence parts and these transcriptional unit parts. So the interchangeable parts, any noun you could plug into the sentence and it's still grammatically correct. And the same thing down in the transcriptional unit, you could plug in any promoter or any operator and it's still consistent.
So we're going to want to make RSC and PBED apart eventually anyway.
Yeah, that's what Roger is suggesting, which is a good suggestion. And that is what they suggest as well.
Okay. I think that's good as long as we know.
So this unit now becomes like the promoter piece. Because this regulates the promoter. Like this is the promoter piece.
So we do a separate isolation of that.
Correct.
when we're doing the GFP one, we want to put it out. Is that what that amounts to? Yeah.
So in the case of the GFP one, I was creating the cassette and the vector, meaning that the cassette is the part that gets cut out and kind of thrown away.
Yeah.
So I wasn't designing a GoldenBraid part at that point.
Right.
But yeah, if we did parts, then it seems like we'd want to separate the operator, as they're showing here, the GAL4, from the promoter.
So then okay, so let's do this suggestion and what Roger suggested. We've decided to lump all operators with their promoters. So that means that when you are designing the vector, you are going to design primers to sit outside of the RC. Let me say that correctly. You are designing primers so that RC is no longer in the vector when you are done with it. So it should be your end result. If you start with, RC, PBAD, GFP, Terminator, or whatever's there, you are putting your restriction sites here and here. And all of our pieces that we make in the future to come together and fit into this. So this is a transcriptional unit that's already existing in this plasmid, right? So in the future, when you are done with it, it's now going to look like you are now going to have inserts of restriction sites on this side, but RSC and PBAD and GFP and the Terminator are still going to be in there. It's just that when you come in with your own thing, it's going to replace this entire thing. Right? So when you come in with your own thing, it now might be CMV promoter, RFP, Terminator, or whatever it is you want. But the RSC regulatory element will no longer be there.
So I have a quick question. So I'm not sure why are you just using this plasmid because you have it? Is that the whole reason you're using this one?
We're designing like a whole repository for
Yeah.
So we're using
Okay, yeah. So this is a two-level system, right? So,
correct.
This is the selection of dapacyllin, and the other one that you mentioned in the paper or in the assignment is the Buck 18. That's also dapacyllin selection. You want to have separate, maybe kanamycin on the omega level or something like that.
Yeah.
But are you gonna? So with this eco, you can use so a negative select. This is negative selection if you use around in the plate, right? Because if it's not inserted, the eco is still it's like the vector was never caught and it was actually transformed. Then you can, as long as you have ravenous, you can do
Exactly.
it.
Yeah, exactly.
All
So
right.
then, like right. So that's what you would also have to have ravenous on the plate to make sure that, like, anything that was not any GFP plasmid that was not replaced with your transcription unit, like you would see that, right? It's only going to be green if you have a ravenous on the plate, and it'll be. Does that make sense? Like they'll all be white if there's not ravenous. So that's only if you're using like RC. And it's the same for other regulatory systems, which we haven't really talked about. It's the same thing with like if you use an IPTG reducible system, right? You have to have like IPTG in the media or the plate in order to see expression of your cassette.
So is it going to be designed that the Puck 18 is going to be the omega level vector?
Right now we have PLoS as the alpha.
Yeah, so I'm just going off the assignment.
Yeah, as the alpha, and then the omega we were going to use that Puck 8, the last C, Puck 18, and then we can always change. So like the paper suggests using, they have like spec cinnamycin I think on one and amp on the other. And we can change out. It would be ideal to change out one of the antibiotic resistances once we've made those vectors, because when you're cloning from one to the other, right, like if you're growing on ampicillin, things that are on kanamycin will die, if you're growing vice versa.
I think it would be useful to use the P-gene P.1. Yeah, okay. What is neomycin?
mean, yeah, you could do that. I mean, if you wanted to, but that's an SV40 promoter. That's not going to do it.
Oh, okay.
And the same thing with the laxi, we would have to have X down replaced to see, you know.
So Nina, one question I had there is when we're talking about the colony selection, LAC-Z versus GFP,
Yeah.
we could actually use the same mechanism, couldn't we, on both the alpha and the omega?
Yeah, for sure.
So that might be easier because we don't have to have two different types of plates, et cetera.
Absolutely. I mean, we could swap out the laxi for RFP or GFP, or I mean, it does not have to be that.
Right.
It absolutely does not have to be that. And
You just swap out the promoter, right? You can just swap the promoter.
and away from PBAT to something else.
All right, no, I'm sorry, but the RSC, you can swap that out. The repressor,
Yeah.
you can like take that out, right?
Yeah, and that's what we're planning on doing. Which was just just 5 minutes ago, which was funny. But yeah, we're going to treat the promoter and its regulatory element as the promoter piece. So that's going to be like every time we put a promoter in that has a regulatory element, that's going to be lumped together as one.
Yeah. But I mean, if you didn't want to have to use the granules, you could just take out RSC, right? And
Right.
you just
Absolutely. Oh yeah, absolutely. So like that promoter would always be on.
Yeah.
Right, because RC is what is responsible for inhibiting that promoter. So if you took RC out, then that promoter should be substituted.
So remember those three categories we talked about?
Yeah.
Should we add one more for parts, or are we too early for that? Is that too ambitious?
Oh,
Like actually designing computers for parts?
I don't think we should do that yet. I don't know.
That was one thing actually, Nina, I was wanting to clarify because the parts, they have kind of alpha vectors or alpha plasmid, omega plasmids, but then they have the parts are stored in a slightly different plasmid because you don't have both BSA and the BSM in that site. It's just the BSA.
Yeah. They have like stuffer vectors, but I don't know I don't know if that's a priority, or what do you think?
Well, if we cut
I don't
out this unit here, how do we store that once you get it?
If we cut out this unit
The,
here, and we
the
put our own transcriptional unit in
ARC,
here,
yeah, PBAD. We're going to cut that out, right?
Yeah.
As that promoter.
right?
Do we care about that? We're going to keep it as
I
part?
mean, we're not really cutting it out. I mean, we're storing this version of it, and then when we put our transcriptional unit in there, we're storing that
Just cut
version
it.
of it.
Okay.
Yeah. Like, I don't know,
So that's fine. We just store it as plasma.
right? I mean, what do you think, Roger?
Yeah, I think when you're talking about one or more TUs, so if you're talking about a TU, it's going to be stored in either an alpha or an omega plasmid.
All right.
Right? And then you might find that you combine a few of them and you might have multiple TUs stored in either alpha or omega plasmid.
Yeah.
Those are all the same and they'll both have, BSA and BSM with different orientation. It's just when for instance, if we wanted to store the RSC operator or operon, whatever you call it, if we wanted to store that, we would then need to store that in what they call a GB part, which has only the BSA cut sites, not the BSM going out, if that makes sense.
Right, so like each one of our parts that we make could be in its own vector.
Right.
Each 15 that's flanked by BSA1 site. But I don't really know.
And
we'll
there might be
see how that pans out, I feel like. Because we might be I think we have to just see how that meets. Because it might be just as easy for us to have a collection of primers, too. And then just, have the vectors that have these parts on them. And just, PCRing up when we need it. And then, saving I don't know. We'll have to see. But it might be worth just making a collection of, at some other point, I think depending on what our volume is and how many products we make, a place to store just the gene that's flanked by the BSA1 site, right? That would make the complementary ends that would be complementary to your promoter on that side and the terminator on the other side. And these are stored in the plasmids with the, or not. I mean, we'll just have to see, I think. Because the GoldenBraid they have like storage vectors for all of their parts.
If you go with the primer approach where you're just storing primers and you PCR it up inside, then you have to do the gel and
then you have to
purify it. So there's a little extra step there? Or
that's an extra step.
Okay.
The more work we do now, the less work gets.
I mean, but the reality is you would still have to cut it out.
Yeah, but that's a single pot for the whole TU.
You cut it
Just a
together,
one-pot
cut it out,
system,
and
right?
one pot it.
Just one pot. Seems like a slot.
I think it'd be interesting, and given that that's what the GoldenBraid does, they have kind of an inventory of those parts. I think it would be an interesting exercise, at least, to create some parts.
Let's have a parts group.
So do they have something minimalistic plasmid they throw the part in?
They have what they call like a stuffer. Well, at least in their GoldenBraid, in the first, I don't know if they renamed it in the, I forget. They do. They have let's see, I don't know if it's in this. Yeah. They have what they call their PPD parts vectors. And they have their parts stored in here, and it's flanked by VSMP1 and HEB1. And this is confusing because I know our system it would be VSA1 and VCP1 or VBS1, whatever. But yeah, they have a vector that they're storing their parts in. It's their universal part domesticator vector.
Okay.
That they take from here and use to put together a transcriptional unit to put in their alpha level vector.
They could just do a one pot by just putting it in the nutrition environment and like it.
So Nina, figure 8.2 is a nice visual graphic again of how they put their parts together. And it shows how you may have different combinations. So sometimes, you have the kind of the operon all the way through the ATG. Sometimes you have that's broken up into smaller pieces, etc. So the very top row, you could do just three parts in one TU.
So theirs is just the promoter, the cysteine, and the terminator. Those are the three main parts.
Right.
And then there are permutations of that as well. So like this would be, I would say that the easiest thing to design at first pass is just a promoter, a gene, and a terminator. 3 parts to put together. With VSA1 sites flanking either or, and they have complementary ends on either side. And they tell you what complementary ends they use, I'm pretty sure. So you could even do what they do in the paper to engineer it. So I don't know if there's something like you want to take apart the lac operon and put luciferase and put a couple of those together. I don't know what.
Yeah, for sure. I guess we could cut out and we could flank luciferase and luciferase as separate genes.
For sure. So what are we thinking? So let's do parts? There's the parts group now, the alpha group, the omega group, and then the removal of the enzymes from the backbone. And then the next stage that we're at, I think, would be thinking about making sure that our backbones have different antibiotic resistances and things like that. But I think this is a one step at a time process, so we're not completely overloaded. I don't know. Are we already completely overloaded? Too late? I mean, are there any other questions before you go off and try to design some primers on your own?
I had one question.
Yeah.
When we're removing recognition sites,
Yeah.
There's different ways that you can do that, right? Based on different coding, et cetera. Do you typically have to find out, is this in an ORF? And if so, try to do it, in a way that doesn't change the amino acids? Or...
Yeah, definitely. That's what I would try to do. Every time you're changing a restriction enzyme site in the backbone or wherever, it would be best practice to change it so that you're maintaining the amino acid sequence.
Okay.
I mean, hopefully your plasmid is annotated and you know where your origin of replication is and you know where the resistance markers are and so forth and so on. But if you don't and there's some ambiguity with that, it's always safe to assume that, I would just assume that you're mutating something that may be important. So, very well annotated.
But you don't know when you're in frame or out of frame, right? Or what frame you're in, frame shift or whatever they call it.
So if you're just mutating a single nucleotide to something else, then you shouldn't be, you don't want to just remove a nucleotide, right? You just want to change it to something else.
Right.
You shouldn't shift the frame.
Right, but you don't know what amino acid you're coding for unless you know what the frame is.
Oh yeah, that's correct. That's correct. So you would need to know where the open reading frame is of that gene.
Okay.
Yeah. So that's why if it's not annotated, if you get a vector backbone and the origin is not annotated or something's not annotated and you're not 100% sure, where that site lies or what it's within, then I would just go ahead and make sure you're keeping the amino acid sequence clean. If it's just in a stuffer region or something in between coding sequences or open reading frames, then it's really, you could probably change it to anything that you want. And you want to make sure that you're not changing it to a site that you are going, you don't want to add a site that is going to be detrimental to your cloning process in the future. You don't want to make a BBS1 accidentally if you're going to be using BBS1 for another step in the cloning. Okay, this is a lot, guys.
Yeah.
This is a lot of stuff.
So, I'm not familiar with the 2.0 version. Do they still just use 4 plasmids?
They're still doing the same thing.
They just blanket with one more course.
Yeah. And I think they're making this into some sort of kit with plasmid. I don't know how far, I don't know where they are, but we are getting some of it, or a lot of it, apparently. Cameron's been in contact with them.
But are there any restrictions from a commercial standpoint?
Well, I don't think we should, I don't think we should be distributing the pieces. I'm pretty sure they're proprietary. I don't know. I don't know, actually, where they are with it. But I'm assuming that there's, IP on it, and they're giving us they're giving us plasmids as a gift for us to try. And they're not exploring the bacterial side of things, so that we're designing, a whole set of these vectors for bacteria. Because what they're giving us are, plant vectors and plant parts. So we're going to take their system and just move it to bacteria and maybe strike up a collaboration with them on that. So that's pretty much what we were why we're designing these, is that we can have them in bacterial expression vectors for all of our stuff. And I think we were talking that if it came down to a point where we wanted to distribute some of these parts, as long as we change something up a little bit, it might be enough to avoid some of the IP mumbo jumbo, but I'm not 100% sure. We will cross that bridge when we get to it.
Yeah, but some of this is already established with the golden game.
Yeah, exactly.
That's open, right?
Yeah.
Open gates and open.
Yeah.
I think it's more open.
I agree. I think so too. So, all right. So what are we doing? What's the consensus? They're all off in their own thing now. Not paying attention. So I don't know. I'll just write alpha, omega, whatever you want to sign up for with designing, I guess, and then do it. So we'll still break up, I guess, into groups. parts, what do we decide? Someone should do a couple of transcriptional units with BSA1 sites and complementary ends. So parts group.
Much upper on as a transcript.
Okay, so you'll do today we'll do Lux, operon parts. So that includes promoter, genes, terminator.
Yeah, I mean, as far as promoters go, I'm not sure what
promoter is on that already?
It's quorum sensing.
Oh, okay.
It's not really a promoter. It's this whole mechanism where the bacteria emits a histone into the water, and then as soon as it gets to high enough concentration, it acts as a promoter.
Yeah. Okay.
But we can't really do that. When you come up with a different, that's why I was just.
So you were going to do the feedback.
I was going to.
So why don't you design? Then you can design feedback, boosted braid genes.
I'll use feedback as a promoter.
Yeah.
I'll use the ARC.
Totally fine.
Okay.
And then alpha and omega, and then feedback. I'm already putting Roger in here because we designed this already. Roger, did you do omega ones too?
Yes, I did.
So anyone can join Roger on that front.
Sorry for stepping up for a second.
You're in trouble. So back down. No, I'm just kidding.
What do you get for me?
What do you want? Omega?
I'll take either.
Alpha or omega?
Either one.
Kyle, what are you doing?
I was just really
No, you don't have to do it. You think about parts you want and distribute them to your group. So Kyle, I'm going to put you down for thinking. Thinking is hard. Thinking parts of the plant.
Of course, no one wants to do the.
Oh, I'll do it.
I'll help if I can.
No, I'll work on
Can I
it too.
I'm just kidding. I'll totally do it. Anyone can join me.
All right, I'll join you.
Yeah, I will too. But I want to learn how to do that.
I'm going to, I wrote people in. Okay.
We like correspond with our group members.
What was that?
I have to correspond with our group members.
Yeah, you have to correspond.
All right, I'll
Does
send
everyone have each other's email
I
address?
should have something.
I
If not, I'll have. We'll ask them for it.
Yeah.
should all be in the group, right? Yeah.
I don't know. I don't have your email. You want to be
My email?
What? No, I have yours.
Okay.
Yeah, I'm not sure.
You don't have to be a part of it. It's
With
okay.
respect to the alpha, I mean, so with respect to another selection marker, why did they choose spectrum monitoring? Was there a prior aspect to it?
Kyle was telling me that a lot of people use kanamycin as a marker in plants. So that's probably the reason why they didn't have it on the backbone, because they're using kanamycin as an actual part in the transcription unit. So I think they ruled that one out. And then
But if you're designing just for the bacteria,
we can totally have that.
yeah.
Absolutely.
I mean, I can put in kanamycin and whatever omega does.
Oh yeah. Okay.
That would be
sounds
cool.
great. Okay. So we'll just put that add kanamycin.
Are we using Puck 18 for that, or?
This one is fucking.
Okay, yeah, I agree.
Yeah.
Does somebody have?
I don't know. Somebody
We have
mentioned-
Puck 18 in the lab,
We
right?
do? I've never seen it, but maybe somebody.
Hold on. You've never seen
Or
it?
is it?
But I don't know much.
You don't know.
Kyle mentioned it, I think.
Kyle, do we have Puck 18 in the lab? I mean, it comes with all these transformation kits. So as like a, you do, yeah. It usually comes as like a positive control transformation kit. So I don't know if we have.
I have one.
We could do Puck 19.
I think it's just the MCF.
Oh, really? Okay. We should check then before we design this.
So Puck 19 is amplification.
Yeah, they're both campuses now.
Okay.
The MCS and Puck 19, if you believe what's on the internet, is first oriented to those of the Puck 18.
Okay.
Yep. Wikipedia is the same thing. So, I mean, if we don't have Puck 18, then I have Puck 19.
I guess we could buy Puck 18.
I mean, I have. Fuck that. I mean, if you're really set on MCS being in that.
No, I don't care.
It's not going to be really.
It doesn't
easy.
matter, really.
Yeah.
We're going to cut it out anyway.
Yeah, so I was going to ask the question, could we not just use the P-glow and change out the amp resistance in it?
Yeah, we can absolutely do that too. I keep GFP as the marker, fluorescence marker. The only thing, let me think, what might get hairy there? GFP.
Well, Puck 19 is a lot smaller. It's only 26K. I mean, sorry, 2K.
Smaller than Vivo.
Oh, yeah.
Vivo's like twice the size.
Yeah.
No, we can use P-glow.
For both alpha and omega?
Yeah. We would just have to. No, we can use P-glow. That's fine.
And then change out so we have what resistance do we want besides amp?
Can. I don't know what's cheapest. What's the cheapest antibiotic?
Can, we have phenylizin.
Do we?
Yeah.
Whatever's next in line to ampicillin, I guess.
Okay.
The cost would be what we would use. So I can't find here. You have can. Let's stick with that.
I think about a walk.
Oh, are you taking it home with you?
We have, no, we have chloramphenicol. I got some of that.
Chloramphenicol is also an option. That might be more expensive. I'm not sure. Well, then why don't we just do it on, we can do both on P-glow. That's totally fine. The alpha and omega on P-glow, it doesn't make a difference. I just had different ones really for practicing making primers, but. So the only difference then between the alpha and omega when you're designing primers for this then is the overhang, right? The annealing region would be the same because you're using the same vector backflow, but the restriction sites are just like flip flop between the alpha and the omega. Okay, so we'll put P-glow. Oh yeah, Roger?
Yeah, but if we do that, we're going to want to swap out one of the amp resistance for something else, right?
Yeah, definitely.
One of them.
So we were thinking Panamaitin.
Okay.
Is there an advantage to using the same plasmid versus having two different ones?
No, I mean not for this, not for cloning purposes.
Okay.
The only time.
The nice thing is if we remove those BSA and BSM sites, we only have to do that once then.
That is correct.
So the advantage is it's simpler. It's just doing one. Okay. That makes sense.
So we're using
What are you?
the amp after the synthesis, and we're putting
Panamaitin.
So you want that done after the restriction enzymes are put into the original vector, the P-glow vector? I mean, I'm just curious of the timeline.
We don't have a timeline quite yet. I think we're just gonna see how long it takes to like engineer the primers and check them with each other, and then we're just gonna pick in order based on people's schedules and what they can do. So if it's like quick and easy to flip out the antibiotic resistance first before we maintain restriction sites out of the backbone and also add restriction sites in, and we can go that way. I think we were just kind of waiting to see how things progress with the design. But there's really no, we can create an order right now, or if you guys want to wait to see who gets done with primers first and people can get started before or digest or whatever.
I think we should get the design down correctly. Make sure that we do that right, because otherwise we can waste a lot of time.
Also, just swapping out, I have to check too, just swapping out the antibiotic resistance, we have to see if that gets rid of any of the BSA, whatever is in the background. I haven't checked where the sites are. So like, BSA1, I think there are BSA1 sites in the backbone of the P-glow.
Yeah, that's in the AMP-R.
That's in the AMP-R?
It is, yeah.
So if we get rid of the AMP, if we swap that out, then we don't have to take that out. Unless there's BSA1 and Panamaitin, which we have to check. The Panamaitin. Are there any other BSA1 sites in that backbone?
No, I don't think so.
No.
Just that one.
What about BSMB1?
Yes, one, but it's in between, it looks like. Non-coding.
So we still would have to get rid of BSMB1.
Right.
Well, and we also need to get rid of the BSA1 because one of the plasmids is going to have the AMP-R, right?
That is true. That is true.
Yeah.
So it looks like Nanolysis has BSMB1 in the Juárez.
Oh, really? Okay, so we can get rid of BSMB1 in the backbone and also in the canteen. And then BSA1 in the AMP-R. Okay. Yeah? See some fear on some faces. All right, if you guys want to volunteer for anything else, but I'll send.
Volunteer later in the game.
You can volunteer at any point in the game.
This seems like pretty good. If we got all this stuff designed, we could order the primers and get started.
Yeah.
Is there a qualitative difference with alpha and omega besides just the way the arrangement? So,
They're just the way the restriction sites are arranged, so you can like go back and forth, right? So you can like clone something out of one and add it. So you can chain multiple transcription units together and you can clone them out and go back and forth.
I don't know if this is useful, but I was thinking we can put up the loop diagram of the first paper, GoldenBraid paper, and I could go through it the way I understood it. If anyone still needs to, does everyone really understand that diagram?
Wait, what do you want? This one? Hold on. The threading?
Yeah, because for some reason it's confusing.
Like this one?
Yeah, the one from the first paper. Does everyone understand it? No?
I'll have to read it one.
Any votes yes? We have 2 no's and several abstain.
Yeah, you want to try it?
I think it would be good to go.
This?
Yeah.
Crazy thing?
It's not that crazy.
Go through it. Do
It's
it.
not that crazy, guys.
Mike,
Alright.
I'll
Alright, so we all understand how the type 2S restriction enzymes work, right?
yeah.
Okay. So there's a recognition site and then it cuts whatever's next. So you can make that overhang look like whatever you want. You get so far, right?
Yeah.
Okay. So if we do that, we can string up words together in a grammar like they talked about in that other paper. So what they're showing here is just overhangs that match up with each other. So this overhang can only anneal to another 4 overhang. Right? So the reason why that's an advantage when you're designing these parts is that they always go in the right order in one pot. You don't need to stitch them together separately. You can stitch them together all at once. Does that make sense so far?
Yeah.
Okay. So we do that, and we put it in A12C alpha vector here. There are 2 alpha vectors in this diagram. Each of these are alpha vectors because they have the BSA1 sites going out from Lexi and the BSMB1 sites going in. It cuts in this direction, leaving an overhang on this side, and it cuts in this direction, leaving an overhang on this side when we use these restrictions. Everyone follow so far?
Yeah.
Okay. So now they're showing how they took these parts and stitched them together and put them in these empty alpha vectors. Okay, and you can use either one. These are both alpha vectors. They're just different in terms of what on overhangs they're using, right?
Yep.
Yeah. So this one, and this, I don't know why they use this weird system for the arrows, but yeah, these two come together, and we've got this part now. See all the parts that come together. They X out the numbers because the restriction sites no longer work. That's what they're trying to signify by Xing the boxes. Everyone get that so far?
Yeah.
Okay. So now we've got a transcriptional unit. So they're turning these individual words. Instead of drawing them all out, they're just calling this sentence 1. So that's what happened between this diagram and that diagram. Really simple. Any questions so far? All right. So now we've got an alpha vector with a transcriptional unit. All right. Now if you look at this point in the diagram, they say the level omega and level alpha entry point. Now all they're doing is showing how you can take a part from the alpha vector and put it into the omega vector, or vice versa. So remember this guy. This is an empty alpha vector. Okay. Now what they're showing is, let's take a look. Okay, so this is an empty alpha vector. This is our alpha vector from up there, right? Okay, so let's start with this one. This is where we were at before. They took this part out and put it into an omega vector with another part from the other alpha vector. Does that make sense?
Yeah.
Let me repeat that one more time. 2 parts, 2 transcription units. They're putting them together in omega by cutting with the same restriction enzyme right there, BSNP1, and opening it up on this side. Notice that these arrows go out. So now we've got arrows going this way, arrows going that way. That's where the overhangs match up. And we've got this here. Okay. Now we can do it again in an alpha vector. So we've got this piece combined from these two in our omega vector. Now we can take 2 omega vectors and put them together in this fresh alpha vector. Does that make sense?
Yeah.
Okay. And then now they're just going back and forth, doing that to make the transcription unit longer and longer.
It's perfect.
So within the context of their GoldenBraid system, you simply have 6 unique core-based overhangs to go. Well, 3 with the
Well, they have 8, right?
So there's
ABC 1, 2, 3.
exactly.
Yeah, okay.
Yeah.
That's true.
So essentially, this is a binary combination system within the GoldenBraid system, right? I mean, I guess the entry vectors give you a little more flexibility for creating transcriptional units to insert into. I don't know. I mean, I guess. At the end of the
All right, thanks for letting me do that. It was
That's
for my
perfect.
edification as well.
That's perfect.
So
My teaching.
That's wonderful.
one other thing that's handy to go through is you just did the left-hand side, but then there's also the right-hand side. So the difference is you either mix it with a, 1AB3, and you end up with the 1,3 part,
Yeah.
or you mix those 2 initial parts with a 3AB2, and you get a 3,2 part.
So this saying you can make 2 parts with the 3 and the 2 here, right? And you get the creation.
So that you can basically start with this one or start with that one. But you're combining them regardless. This is putting them in different omega vectors. So here they put these 2 parts in this omega vector on this side, and on this side they just put these 2 parts in this omega vector.
Right, so it's kind of like each time you do one of these reactions, you can decide, do I want the combined product to be a left product or a right product?
Makes sense. You know what? It would have been so much easier if they actually label this as TU2, right? I mean, that just kind of seems like common sense.
Right.
Yes, it's a little confusing with their
Numbering system.
Yeah.
Let's fix
Well,
it.
they were consistent with their typo.
That's true.
That's
I don't know what it is, but that's fine. So you can put them in either order is what the end result is.
So what's 3 and 4?
What's 3 and 4?
You mean TU 3 and 4?
Yeah.
Yeah, it's just like you said. It's in the first one they put 2 1s together and somehow showed a 1 and a 2 as the result. And
Yeah.
the next layer they put 1s and 2s together and you got 3s and 4s somehow.
They arbitrarily labeled one of the 1s and 2s as these.
It's really like T1, T2. It's really TU1, TU2, TU1, TU2. But they changed
Right.
it at the bottom.
Right.
Yeah.
Yeah.
And they should have.
Well, actually, Nina, it's actually T1, TU1, 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 TU1.
Oh, right. That's true. That is true. From the beginning, it is all TU1. Yeah. Maybe it is. Maybe it's just like repetition of the same coding sequence over and over again.
Because that makes perfect sense. That's the thing we want to do.
So, thank you. That was awesome. Thanks for going through that. All right. So we all know what we have to do?
So just to clarify from I would say, so
Yeah.
you can go either the left-hand side or the right-hand side with the same construct, right?
Yes.
And which side you go to depends upon whether or not you want the 5' or the 3' position of what you're combining.
Right. Yeah. Which is totally confusing how they documented it. Yeah. But, yeah. Okay. So, I'm overloaded. Are you? I think. Okay, so we all know what we have to do. And we will keep each other posted on our design and check each other's primer design.
So at the next meeting, do you want to go through them then, or what do you want to do at the next meeting?
What do you guys think? Let's ask them what, at some point, the plant scientists, they kept on saying they wanted to like take over and talk about something, but I don't know when that's happening.
Oh.
So if that's not next week,
2
then
weeks.
for 2 weeks, then we'll go through primer design, I
Okay.
guess. I mean, if it's 2 weeks, is that enough time for people to?
Can we do some of this via email correspondence?
We're gonna have.
Oh, absolutely.
Yeah,
We're gonna
so-
have
Yeah,
to talk
yes,
with
yes,
each
yes.
other. We're gonna have
Like we should send
Most
each
of
other
it's
files,
nice.
and they should be well labeled with what you have designed and what part of the vector and where the enzyme sites are and what orientation they're in. Like I don't want to just see a sequence of letters. Like it needs to be what's the 5' to 3' orientation.
Awesome.
It's more like programming. You gotta write comments.
Is it? I
I don't know.
don't know. But yeah. If we're getting so corresponding via email, but you have to be really descriptive when you're sending someone else your design, so they know what they're looking at.
Great.
Okay. Awesome.
Are you gonna
You're
put out
looking like
an
good
email
project.
just to give us the abstract, read abstract of what we're doing?
The abstract.
Not the abstract, just
Oh, you mean the overview?
yeah.
Okay, I'll break it down. The breakdown is that we need to design the alpha and omega entry vectors, right? And we decided that we're going to do alpha and omega both on PLoS. But we're going to make 2 varieties. The alpha PLoS is going to have amp resistance to the backbone, and the omega PLoS is going to have pan. So one's going to be amp and one's going to be pan. So that'll be great. We'll have both GFP, which means that when we put a transcription unit in, GFP should be out, and our stuff will be in. So we should go into 3 colonies to white colonies. Okay, so alpha and omega in PLoS, one should have amp and one should have pan. So we have divided up the labor so that we can change on one vectors to amp to pan. We also divided up the labor so that we can engineer primers to mutate out BSMB1 and BSA1 sites that are present in the backbone, because we don't want to be digesting our transcriptional units with BSA1 and also be chopping up our vector. That would be awful, so let's avoid that. So we'll mutate those out. And then we divided up the labor so that we have some parts to play with. So these parts will be flanked by BSA1 sites, and they'll have complementary ends. So your promoter, your 5' ends of promoters will be complementary to the vector. The 3' ends of promoters will be complementary to the 5' end of your beam, and vice versa.
So what's the plan of action for removing those? They are mutating those sites.
Yeah, I was going to do like inverse PCR.
Okay.
Yeah. So just have a primer that goes in this direction and changes the site, and another primer in this direction, and we amplify around. I might do EIPCR, which puts enzyme sites on the ends of these so that when we make the double strands, we can digest and ligate.
Usually it's one phone, right?
That is true. Yeah. I can do that too. So that would just be inverse PCR, where their front ends would be, I mentioned earlier, and then we would just ligate.
Are you gonna jump your pod? Yeah, or just do like
Yeah, purify the product. But then, so that's gonna be like, if we had to change 3 sites and there, we have to do this, we might have to do this multiple times. That makes sense.
Even if all you need to do is change one nucleotide, right?
If we just have to change one site and one nucleotide, then that's one reaction.
But since we have multiple sites, we have 2 sites, so have to do it twice.
Yeah, where we can throw multiple pairs of primers into the mix, like I've done that before. So it reduces the efficiency, but
Get some product.
I don't know if you've done it that way before. I've done like site directed, where I've mutated like sites in different locations, and I throw all 3 primers into the mix, and it works great.
like the one you're mutating, like BSD1,
Yeah.
BSA1. You can also do BSD1 digest food related to it.
Right, so like after you do this, you want to DPM1. The DPM1 digest will digest all of the template, because that was extracted from bacteria, so it's methylated. So we can digest all the template, and that would be like the unmutated form, so that we will hopefully only have our product. And so when we're transforming in, it should increase the frequency at which we're obtaining vectors that have our mutation incorporated in it, as opposed to just getting back down on a wild type. That sort of makes sense.
So that the DPM1, you said?
Yeah. So like our plasmid that we've isolated from bacteria, so like let's say we're PCRing off of a mini prep or something, like that DNA is going to be methylated. So, and our PCR products won't. So DPN1 selectively digests methylated DNA.
Oh, okay.
All right, are we good? Ish? Ish?
It's the hard part. Right?
What did you say?
Now we actually have to do the hard part.
Yeah, you do.
Thanks, Nina, for another rousing IdeaLab.
You are welcome. I like to give out more work at the end of every IdeaLab, so that is not the intention when they start. I just want to get that out there. That is not the intention. Okay.
It's sort of an escalating thing where by the end of the year, we just have
Now we have like so many projects we don't know what to do with. All right. any other questions, comments, concerns before we close? We'll all communicate via email. All right. Awesome. Okay. Wonderful. Thank you.
I just hope you're prepared for the deluge of emails you're gonna get.
I'm not ready for it. Give me like a couple of days.
Bye, guys.
Bye, Roger.
Bye, Roger.